Now we're going to talk about a concept called capillary exchange. It's kind of a complicated process, which is why I'm going to be doing some diagrams to try to explain it to you. I encourage you to take out a piece of paper and do the note together with me. So capillary exchange, what exactly does that mean? Basically means the exchange of materials across the capillary walls. So what goes into the capillaries, what goes out of the capillary. Now that actually is going to be dependent on several factors. We are going to be looking at three of these factors. So the first factor that affects capillary exchange is just simple diffusion. Right? So diffusion, as you know, involves moving the movement of material from an area of high concentration, that's what these square brackets means, concentration, to an area of low concentration. Okay, so suppose we have a capillary vessel here. Yeah, let's just draw the capillary right here, and for the capillary, we are going to have one end that is connected to the arterial. So we call that the arterial end. Arterial end. Okay, we're going to have the other end that is going to be connected to venules. So we call that the venous end. Okay. And in between is where you will find the capillary. As discussed previously, the capillary wall is quite leaky, so I'm going to put some holes here. So you know, a lot of materials are actually free to move in and out of the capillary walls, and a lot of them do so by using diffusion. So if this is a systemic capillary, which means the outside here would be some kind of tissue, right? So it could be like the liver, it could be the kidneys, whatever it is, and here it would be just the blood that is fresh from your pulmonary circuit. So the oxygen content within the blood here is going to be high. So we have high O2, right? Compared to the tissue outside, it's going to have low O2 because the tissue is using the oxygen, right? So because of this difference in concentration, then the oxygen is going to come out. Okay, so you're going to have movement of oxygen from the inside of the capillary to the outside. Contrary to oxygen, the carbon dioxide level in the arterial blood is going to be low because it's fresh from the lungs, and whereas the outside here, you're going to have high carbon dioxide, because once again out here what you're doing is you're taking glucose, right? You're reacting it with oxygen, which then produces carbon dioxide and water. So you're going to have a lot of carbon dioxide accumulating in the tissue, right? So what happens is the carbon dioxide is going to go from high concentration to low concentration. It's going to go in, right? And afterwards, in the venous side, we're going to end up having high CO2, and we are going to have low O2. So of course, this blood is going to eventually go back to lungs, where you're gonna reoxygenate it and then have another go at it. But basically that's what the first factor is, right? In terms of controlling what goes in and out, there just has to be some kind of concentration difference, and if the molecule is able to cross the capillary walls, then it's going to do so. Sometimes the molecules are too big, they cannot fit through the gaps, then they won't be able to move using diffusion. So I'm just going to give you another example, right? You might have high glucose here, high glucose, and then you will have low glucose on the outside, and again it would just go from high to low concentration. So that's the first factor. It's quite straightforward, right? In terms of controlling which way things are going to move, right? Just from high to low, as long as they can fit through the gaps. So the next factor that affects capillary exchange that we're going to look at is blood pressure. Okay, so blood pressure is basically the amount of force that the blood is pushing against the blood vessel. So you can think of a water tap. Okay, so here's my water tap. Okay, and then I'm gonna hook up a hose to the water tap. Okay, and before I turn it on, I'm gonna poke some holes in the hose here, and then I'm gonna turn on the water tap to the maximum. Okay, so imagine, visualize what's gonna happen, right? If you think about this, the water will shoot out quite high, but then eventually, as you go farther away from the water tap, the water is still going to be coming out, but it's not going to be shooting out with as much of a force as before. That's because the farther away you are from the source here, the lower the pressure will be, and so there's not as much force pushing the liquid out. So in this analogy, the water tap is equivalent to the heart, and this hose is basically your blood vessels. So when you're in the aorta, right, which is directly connected to the heart, blood pressure is really high, right? It will shoot the blood out. But as you go to the arterial, right, the capillary, the venules, and eventually the veins, what's going to happen is the pressure is going to decrease. So blood pressure is going to be the highest when it's closer to the heart, right? And it will be lower the further away you are from the heart. So let's look at our capillary again. Let's draw another new one. There is my capillary, and as I mentioned before, you're going to have the arterial side and the venous side here, and then you're going to have the tissue here. So what happens is the blood pressure will be responsible for pushing fluids out of your capillary. Okay, it doesn't push the blood out. Okay, the blood remains inside the vessel. You're pushing out fluid. So we're going to learn more about the composition of blood in the next lecture. But basically, these fluids are like mostly water with some dissolved minerals in it. Okay, we measure blood pressure using mmHg. So just for reference, we will say the blood pressure here is going to be 30 mmHg, and over here on the venous side, the blood pressure is going to be, say, half of that. Okay, 15 mmHg. Right. So the key thing here, two key things here to remember are these. Number one, the blood pressure BP, right, is responsible for pushing, right, pushes fluid out of the capillary, right, and then secondly, the BP on the arterial side is going to be greater than it is on the venous side. So you'll be losing more fluid on the arterial side than you would on the venous side. So that's the second factor affecting capillary exchange. Let's take a look at the third factor. Okay, the third factor. The third factor that affects capillary exchange is what we call osmotic pressure. Osmotic pressure. Okay, so to understand osmotic pressure, let's take a look at this example. And let's say I have two chambers, and they are going to be separated by a semi-permeable membrane. Okay, semi-permeable membrane, and this membrane is quite it's not very porous. The gaps are very small, so only water can pass through. Right? So let's think about this. Let's say I have some solutes here, some molecules. Okay, so these are solutes that are dissolved in the left chambers, and then I have the same solute that are dissolved on the right chamber. Right? So over here we have a lot of the solute. Right? So we're going to say high solute concentration. Okay, and over here we're going to have low solute concentration. Right? So if there is no membrane, then what's going to happen is the solutes, the pink circles here, they are going to go from high concentration to low concentration. They're going to move from left to right. But here's the thing, we put a membrane here, so even though these pink circles want to go to the right side, down their concentration gradient, they can't, because they won't fit through the membrane. So the only thing that can move here is water. Right? So water is going to move from this side, from the right side, towards the left side, right? Water is going to move from low concentration to high concentration, right? This is called osmosis, right? You might remember that from last semester, right? So the water will move to the left here to try to dilute out the concentration and balance it out. Okay? So another way to think about this, if it's a little bit confusing, is if you take a spoon of salt and you put it in your mouth, right? Then it literally sucks the saliva out of your mouth, right? So the salt doesn't really go into the cell of your cheeks, for example, but rather it creates an osmotic pressure that will suck the fluid out of the cells instead. So that's what happens here. So keeping that in mind, let's take a look at what happens in our capillaries. Okay, so I'm going to draw another one over here. So inside the capillaries, we are going to have a lot of dissolved stuff. Okay, so we have blood cells, for example, blood cells. These are red blood cells. We are going to have dissolved electrolytes like potassium, sodium. We're gonna have proteins in it. So that's what these pink dots are. Okay. So these are all dissolved in the blood. They're not going anywhere, right? The proteins are too big to leave the capillaries wall, and the cells are too big to leave the walls. Okay, only fluids can move through, right? And then gases. On the outside, the tissue, we're also going to have some of these things, but typically speaking, the concentration is much lower, right? So what happens is this is exactly like the situation we talked about on top over here, right? Whereas the left side is actually the capillary and the right side is actually tissue, right? So we have more solute dissolved in the blood than we do in the tissue, and as a result, that creates osmotic pressure that would suck fluid into the capillary. Okay. It would take the fluid from the outside and draw it inside. Oops, sorry, let me fix the arrow here. There we go. Now, this osmotic pressure created by solute concentration difference across the capillary wall is going to be the same from the arterial side and the venous side, because the pink dots, right, is pretty much uniform from one side to the other, right? So this is very different than the blood pressure. Okay, blood pressure, as you go from arterial side to venous side, they tend to decrease. However, for osmotic pressure, that remains unchanged. So OS, osmotic OP rather, sorry, osmotic pressure will draw fluid, right? Draws fluid into the capillary, as opposed to blood pressure is going to be pushing fluid out of the capillary. Okay. So these two forces, of course, are going to be opposing each other, right? So let's put them in one single diagram and see how it's going to play out. Okay, so I'm just gonna take this one, copy it, and gonna add the green arrows to it. All right, so the blood pressure over here, as I said, is going to be 30 mmHg, right? mmHg. Now we're gonna have the osmotic pressure counteracting that, and it's going to be pointing inwards. Okay, and remember, even though blood pressure drops from arterial side to venous side, the osmotic pressure is going to remain unchanged. So the size of the arrows are going to be the same. All right, and I'm gonna give it a number to help you understand the situation. The osmotic pressure is going to be about 20 mmHg, and over here on the venous side, the osmotic pressure is also going to be 20 mmHg. Remember, it remains constant. So if you take a look at this, you have 30 units of pressure pushing fluid out, and you have 20 unit of pressure pulling fluid into the capillaries. So if you add them up, you would have a net pressure of 10 mmHg. And because the red pressure is greater than the green one, the blood pressure is greater than the osmotic pressure, you will be having a net pressure of 10 mmHg out. That means on the arterial side, you will experience fluid loss. Okay, so this is fluid loss. On the other side, on the venous side, we have 15 mmHg pushing out, 20 mmHg pulling fluid in. If you do the calculation, we have a net pressure of 5 mmHg, and this time the green is bigger than the red, so this is going to be inwards. Right? So as opposed to fluid loss, on the right side, you're going to have fluid gain. Right? So over here, BP is less than OP. But you can see the net difference, net pressure, is greater on the arterial side than it is on the venous side, which means you are actually going to be losing more fluid than you are able to bring back in. So if this keeps on happening, you're going to have fluids accumulating in the tissue. So there is something called the lymphatic system that we'll learn later on that will help collect these extra fluid from the tissue and return it to circulation. So we're going to learn about that later on in the in another lecture. But for now, that's basically what capillary exchange is. Back to the PowerPoint, there are some concept check questions that you can check out. Please do them first before you watch the rest of the video. Which of the following is correct about arterial side of the capillary bed? So on the arterial side, which is over here in the diagram A and V, the BP is going to be about 30 mmHg, whereas the OP, osmotic pressure, is going to be about 20. So BP is greater than OP, and the net pressure is going to be pushing fluid out. Which of the following is true about osmotic pressure of a capillary bed? So osmotic pressure is created by the solute concentration difference, and that difference is pretty much the same on either side, the A side and the V side, and as a result, it will be constant throughout.